Hi, everybody. All right, so in the grade book, you notice that there's the column that has the exam one grades, right? So currently it's hidden. So there's a couple more makeup exams I need to do this afternoon, but as soon as they're done, I'm going to show the column, and then you'll be able to see your grades. So there's a range of scores. So there weren't any like really low scores or anything. But the range is from 40 to 100. So, the average is 74. So, and that was pretty consistent across all four test versions. So that's a good sign. So, and I looked through already. There aren't any questions that nobody got. I think the lowest percentage where people got it right was 30%, which is okay. Some questions were harder than others. So anyway, so that's what you'll see later. You'll just see the score in the thing there. I think tomorrow or sometime this week, I need to enter a how you're doing currently grade for the university for a grade check. So if you're on the lower side, you might be getting an alert about that, because I think it alerts people who are on D and F. But there's still plenty of time in the semester. So the main thing is if you do have a score that you don't love, then you've got to try and work out how to get that up, right? So sound good? Any questions? No? Everyone good? Okay. All right, so that refers to Unit 1. So now we're starting Unit 2. So Unit 2 is going to be talking about the integumentary system and the digestive system. Right, so it's fewer lectures. I've posted all the lectures already, so you can take a look at those and look ahead. I might post a couple of extra little things of additional images. So you notice that one's gone up for skin already. So those are just extra kind of practice images that, again, if I dip into virtual pictures, then those would be good ones to look at. So you don't have to trawl the internet to find alternate images. Right, so that should all be fine. Okay, now the nice thing about integument for humans is it's not quite as extensive as it is for a lot of other animals. So if you were doing comparative anatomy, for example, you might also be looking at feathers or scales, as well as hairs and skin. And layers of skin and that sort of thing, right? So for us, what we're mainly looking at is the layers that make up the skin, the cells that you find in the different layers of the skin, and then the accessory structures that you'd find in different areas of skin, right? And skin around different parts of the body is going to look slightly different, right? So when we talk about the oral cavity, you'll see the skin over the lips is significantly different than the skin that you find kind of in the areas surrounding on the rest of your face. And that skin looks a little bit different than the skin that you'd find on your armpit, or the skin that you'd find on the palm of your hand or the sole of your foot. Right, so it's those sort of differences and being able to recognize what structure you're looking at where that you want to focus on. So, but it should be a pretty nice linear sort of unit. And then we're coming back to eye towards the end of the lecture to look at things like eyelid and some of the other parts of the eye. So, and as you're thinking about these things, it's good to remind yourself what some of the general functions of skin are. Right? So probably one of the biggest ones is protection, because it is one of your innate barriers from the outside. So, not only stopping microbes and things and foreign particles getting in, but stopping yourself losing too much fluid, for example, right? So dehydration, right, is kind of controlled by sweating in humans, for example. So that's one sort of thing there. In your ears, you've got earwax, right, which is a protective measure, stopping, again, insects and things getting where they shouldn't be. Unless you dig really deep on YouTube, you can probably find some things where some giant bug crawled into someone's ear or something, but normally, that doesn't happen. So and different layers, might be more abrasion resistant in certain areas of the body than in others. Again, temperature regulation, there's going to be lots and lots of capillaries and larger blood vessels in different areas of the skin. So all the things that we looked at in Unit 1, the epithelium, the connective tissue, those luminal structures and the structures that aren't luminal, being able to recognize those things is going to really help you out in this unit for both integumentary and digestive systems. Right? So if there's stuff from Unit 1 that didn't make a lot of sense, it's probably not a bad idea to try and identify that so that it does make sense this time. Because you'll need to recognize types of epithelium to recognize what structures you're looking at, like whether it's a duct or a secretion part or some other thing. There's going to be a few different sensory structures. So 2 mainly in the skin that we'll look at, but there are other ones there. They just don't show up very easily, either because they're like ridiculously rare or because they're ridiculously tiny. But nerve bundles, you want to be able to recognize one of those when you see them. Different types of corpuscles, they only occur in certain places. So you kind of want to get to the point where you kind of expect to see certain things in certain places, and then you look for what you expect them to look like, and then you find them. So it's a self-fulfilling prophecy, which is the best type of prophecy, really. So that's what we've got there. Again, for excretion, sweat is the main thing we're talking about. There's also like oil glands, but there'll be a lot more commonly sweat glands in any type of skin that you look at. So you can't really see much evidence of relationship with vitamin D, but it's something that's happening in the skin, but it's not really something unique you're going to look for physical correlates for. Right, so this is kind of showing you how the gross anatomy relates to the histology. Right, so you're not going to see too many images like this, but if you've been recently skinning an animal, you might have been identifying the subcutaneous area, right? Because that's the easiest way to separate the cutaneous membrane from the underlying deep fascia, right? And so when you look at this picture here, what you can see is this outermost layer and the connective tissue layer right next to it. Those are called the cutaneous membrane, right? So that's the epidermis and the dermis. Epidermis is epithelial. Dermis is mostly a connective tissue zone. Right? Underneath the cutaneous membrane, you're going to have the subcutaneous layer, which is also sometimes called superficial fascia. And that can be pretty rich in adipose cells. Right? It's a fatty tissue, especially in mammals. So that's what you're seeing with this kind of yellow layer here. This layer here, this first gray layer, that would be what we call the deep fascia, right? The fascia that sort of surrounds muscles and surrounds muscles in muscle compartments, right? And that's a lot more dense than what you're looking at, right? So in some images, you might see like a dense connective tissue. That would be maybe part of that dense fascia. It could be a tendon, that sort of thing. Right, so that's what you've got there. If you look at this picture here and try to translate that across, the purple area here, so this would be the surface on the lower edge, and then this purple would be the epithelium. So in the case of skin, it's going to be a stratified squamous epithelium that is keratinized. Right? And then underneath it, this pink, that's going to be the dermis. When you zoom in closer, you'll be able to pick out a papillary layer, which is the smaller, closer to the surface layer. And then you've got this kind of thicker pink layer, which is going to be the reticular part of the dermis. Right? So we're going to go through these in other slides, but just so you can see it all together. Right, so epidermis, dermis, most of this, all of this white here, that's going to be the hypodermis, which is that subcutaneous layer. If you were to zoom in on this, you'd be able to recognize that as dense regular connective tissue, so some sort of deeper fascia. And then this area you'd recognize as an underlying skeletal muscle. Right? So a lot of those features you can't see when you're zoomed out like this. But it's like a house, right? There's certain ways that you put, like, siding, insulation, bricks, wallpaper. There's a certain order that those things go in, right? You don't put wallpaper on the outside of the siding because it would be ridiculous, right? You don't put the insulation just on the very, very inside next to the couch, that sort of thing, right? There's an order to how things are arranged. And it's the same with these tissues, right? Whenever you've got epithelium, you're always going to find a connective tissue next to it. As you go deeper, you're going to find more robust, thicker connective tissue, and then you're going to get to some of those other structures. So, and if you were going to look for a blood vessel or a gland or a nervous structure, you'd be wanting to look in this kind of pink or white layer. And there'd be lots. So that's what we're going to do. We're going to zoom in. Also, when you look at this here, and you can see that the epithelium is really thick, right? And again, thick is a very relative term, right? Once you've seen a few pictures, you'll get a pretty good idea about what looks thick and what looks thin, right? And so for this one, the epithelium is mostly stratum corneum, which is going to be the outermost layer, right? The layer of dead cells, which is actually made up of multiple layers of cells. So what I'm talking about with those stratum terms is the actual sublayers of cells that make up the skin, right? The epidermis, right? And as you go through and look at these in different examples, what you'll notice is, depending on where they're from, you might see 4 layers, which will be the case for most of the skin over the body, or you'll see 5 layers if you're in the palms of the hands or the soles of the feet, right? So when you've got 5 layers, we call that thick skin. And when you're in any other area, we call that thin skin, right? So if you take a hand, on the anterior surface, that's going to be thick skin. On the back of the hand, it's thin skin, right? So thick skin tends to have a lot more sensory structures than the thin skin, right? So when you touch something, you don't necessarily use the back of your hand to do that, right? You use a fingertip or something. Okay, so these are the layers, the strata of the epidermis. So going from the outside, you can see the stratum corneum, which is the kind of horny, cornified layer, which is very, very pretty much just keratin, right? There's no real organelles in these. There's no nuclei. It's just keratin, right? The protein, right? And so you get these things here. They're very, very flat, and so they get kind of squished so that it's actually hard to pick out individual cells at some point. You also see these little ones kind of floating off because we're always losing skin cells, right? We're always like shedding skin. If you don't, exfoliate for a while, it tends to build up, and then, it's not ideal. Right, so that's stratum corneum. And then underneath it, you've got stratum lucidum. So stratum lucidum, you only find it in thick skin, but it doesn't always show up, right? So when you see it, it's going to be like just a few cells thick. So it's not a very thick layer, but it's a few cells thick, and it always has this kind of like clear, kind of glassy appearance, right? There's never any big granules or anything in it, right? And it's handy because it's right next to this other layer, which is about the same thickness, called the stratum granulosum, and that is nothing but granules, right? So, and you can see here, there's nuclei here, and there's not nuclei here, right? So these cells in stratum granulosum are just filling up with these granules. The cells are dying off. They're getting rid of their nuclei and all that sort of stuff, right? They're just dying off, becoming even more flat, right? And so cells are produced in this bottom layer, which is the stratum basal, or basal. And then it sounds classier if you say basal. You see, it's all about how it sounds. So that's this layer here. And so this layer here at the very bottom, that can look like it's kind of columnar cells, right? Some look cuboidal, some look columnar, but they look really different than the surface cells. But it's the surface cells that are used for the naming. So it's a stratification of flat cells at the top. Right? So all of this is stratified squamous epithelium, right? Which is going to be different than other stratified squamous epithelium you see, like the non-keratinized types. They don't typically have strata. They're just a thickness of cells, right? But skin's different. It's got layers. So in between stratum basal and stratum granulosum, you've got this really thick layer. So in thin skin, the thickest layer is stratum spinosum, which is this layer here, right? And it's called stratum spinosum, as you remember from Unit 1, because it's got these desmosome junctions giving it a spiny appearance to the cells, right? So stratum spinosum. So you've got the basal layer, the spiny layer, the granular layer, the glassy layer, or clear, and then you've got the horny or cornified layer. Okay, so that's the order, right? They always occur in that order. There's a little mnemonic that I like to use that's "come, let's get sunburned," because who doesn't like a bit of dark humor, you know? So corneum is come, lucidum is let's, granulosum get, spinosum sun, and then basal burned, right? So it shouldn't be too tricky to remember the order of those layers. Next, you want to remember which cells are associated with which layers, right? So at the top, so 90% of the cells in skin are going to be what we call keratinocytes, right? And so they're going to be the general type of epithelial cell that you find in skin, right? Skin cells, right? When you get to the top, right, sometimes we call these corneocytes, right, because they're in the corneum, right? So that's what you get here. Another term that you might see for the ones which are kind of like shedding are called squames, S-Q-U-A-M-E-S, right? And so that's referring really just to these kind of top ones, right? You can see lamellar granules in these keratinocytes in the granular layer. And then you've got some special ones in these lower layers, right? So these intradermal macrophages, so you can also call those Langerhans cells, right? So those are immune system cells, which are kind of inside the skin, kind of functioning a little bit like macrophages or dendritic cells, right? They're engulfing things, they're kind of reporting to the rest of the immune system, that sort of stuff. Right? But you mainly find those in the stratum spinosum. The cells that you're mainly going to find in the stratum basal, so you're going to find stem cells, right? So if you see cells that are going through mitosis, it's probably going to be in the stratum basal. You've also got touch-sensitive cells called Merkel cells. These are also called tactile cells, and those are always at the bottom. Technically, they should be attached to a neuron, a sensory neuron, but you don't in practice ever really see the neuron attached to it. You just see a little cell. And one of the problems with that is that little cell is going to look like a nucleus with a little like halo around it, right? Which is going to look different than some of the other cells. Another cell which is going to have basically the same appearance is going to be the melanocyte, right? So the melanocytes are the cells which are producing the pigment. And in practice, melanocytes look really different to tactile cells, because where the tactile cell is like a disc, like a tactile disc, the melanocyte is actually going to be having all these little extensions, these finger-like extensions that kind of go in between the keratinocytes next to it. And when you see it, so you think like melanocyte, ooh, pigment, lots of melanin, pigment, right? So you'd expect it to be, a darker looking cell, but it's not, because what it's doing is it's producing the pigment and then giving it away to all of the surrounding cells, right? So when you see skin that has some sort of, melanistic pigment, it's usually the cells around the melanocyte which are going to be colored, right? And there's a couple of examples of that you'll better see. But again, in the light microscope, when you're actually looking for these things, any of those special cells pretty much just look like, nucleus that has like a halo around it. So they don't look that different on the slides that you're going to be mostly looking at. All right, so that's pretty much those things. At the bottom, there will be a basement membrane. So between the epithelium and the connective tissue, there will be a basement membrane, but it's not a particularly thick one in skin. It's just sort of there because it has to be there. And then immediately next to that, you're going to have the papillary layer of the dermis, right? So if you imagine this, the skin isn't just two layers on top of each other like, icing on a cake, right? If you were to try and make a cake that was like skin, what you'd want to do is make little divots in the top of the cake, and then you'd want to have some of that kind of icing going down into it. And so those things going down would be the epidermal ridges, and next to them would be the dermal papillae, right? So it's like hills and valleys, right? And part of the reason for that is to increase the surface area because you need diffusion to happen across that boundary, right? It's also there to kind of give a really secure kind of hold between those two layers, right? You don't want those two layers separating. Just like between these other cells here, right? You have lots and lots of desmosomes connecting the cells together. There's other junctions which are connecting the cells together. And all of that is leading to the properties of like waterproofing or, stopping things penetrating through the skin. So here's another little picture just showing the same thing. The text on the side of the screen, it's usually there for like a reason. So it's summarizing something. So most of what I'm saying should be covered on the side there. So, if there's something that I say that isn't on the side, you can always ask me about it. But most of the things should be covered. Like this little piece here, I didn't mention elidin yet, right? But elidin is, the kind of chemical which is mostly found in that stratum lucidum, right? And it's going to be one of these intermediates. Because you don't just go straight to keratin, you go through intermediates like keratohyalin and elidin, and then you get full-on keratin, right? So, it's a process of keratinization is what it's called, right? So, but like you would have seen in Unit 1, most of the bases are covered on the left. Okay. So far, so good? All right, so you want to be able to tell apart, like if I showed you a picture of a skin specimen, you want to be able to tell pretty quickly whether it's thin skin or thick skin, and then you could make an intelligent guess about where in the body it could have come from, right? Because thick skin is only found in certain places. There are certain glands in some areas of thin skin that are only found in certain parts of the body, right? So you can make some pretty intelligent guesses. If you look at the number of structures on this picture on the left versus on the right, you notice that there's a lot more on the left, right? And that's because these accessory structures, things like hair and all the structures that come along with hair, so we call them pilosebaceous units, right? Because it's a hair, a sebaceous gland, and a muscle called an erector pili, right? And so that erector muscle is the muscle that gives you like goosebumps, right? It's attached to the follicle, it's anchored to the rest of the skin, and it just pulls to kind of pull the hair up, right? Which in some animals is super useful because it traps lots of air and creates insulation, or it just makes you look bigger and scarier. In humans, it's there, making up the numbers, So that's what you see there. So in thin skin, you would expect to see hair. In thick skin, you would not expect to see hair. right? It's going to be glabrous or not hairy, right? So you can see here there's an actual hair. The hair has multiple parts, so you can talk about the hair in terms of parts that you could potentially grab if you were like tweezing, or you can talk about parts as in layers, right? So we're going to do both. So the piece of the hair which kind of pokes out from the skin, that's called the hair shaft. The piece of the hair which is underneath the skin, kind of within the skin, that's called the hair root. And then at the bottom of the hair, you've got this expanded bulbous piece, and that's called the hair bulb. Right? So you've got the shaft, the root, and the bulb. And those are going to have, like I said, layers, but there's other pictures that show the layers. Usually, you find sebaceous glands, oil glands, right up against the hair follicles. Right? The hair follicles are the kind of infoldings of the dermis and epidermis, which kind of surround the hair, protect the hair, and give nutrition to the hair. Right? If you imagine like having like layers of the epidermis and dermis, and then you folded that down, right? Those layers which are named this way would have the same sort of relation as the cell's layers which are this way, right? So the follicles go down for the epithelium, and then at the bottom you have a plug which sticks up into the bulb, and that's where you get the blood vessels coming in to give nutrition and stuff to the stem cells which are in the matrix region. So if you look here, you can see this little oval area, right? That's called the hair papilla. That's where the dermis is poking up, and then the rest of this is epidermis derived, right? So you're going to see some better pictures of that in a minute. But if you see hair, it's a surefire sign it's thin skin, right? You'll find sweat glands called eccrine sweat glands in both types of skin, thin and thick, right? So, this is the kind of, regular type of glistening sweat that, you don't even notice sometimes that you're kind of like having sweat. It's called insensible sweat, right? When you notice that you're sweating, that's called sensible sweat, right? Because you're like aware of it. And by that point, a lot of things are sweating probably, and, So, but there's a lot of these eccrine sweat glands, right? If you get sweaty palms, that's eccrine sweat glands, right? So you can see those over here. So these are the kind of sweat glands that we've looked at so far. Like in Unit 1, we saw sweat glands. These are the simple coiled tubular type things, right? If you look in like the armpit region, you'd find a larger type of sweat gland called an apocrine sweat gland, which still uses merocrine secretion. It's just called an apocrine sweat gland. And it's a lot bigger, and those sometimes empty into hair follicles. The eccrine sweat glands never empty into hair follicles, right? So there's lots of little clues you can look for. When you see the two side by side, they look nothing alike, except for being coiled and tubular, right? In thick skin, it's going to be a lot easier to see sensory structures. You're going to find one type of sensory structure in the papillary layer, and you're going to find one type of sensory structure in the reticular layer. So you can kind of see here, there's one corpuscle up here, Meissner's, and there's one corpuscle down here, Pacinian, right? So those are going to be the two major sensory structures that you'll want to look for. And there's lots of blood vessels here. Here it makes it look very, very neat. We've got this plexus between papillary and reticular dermis zones. And then you've got a plexus kind of, between the dermis and the hypodermis. That's great. And yeah, those things are definitely present, but it's not always going to be quite as pretty as that. You'll notice, though, that as you get closer to the surface, you see more capillaries, right? So the collagen fibers are going to be more delicate and more open framework because it's an areolar connective tissue. As you go deeper, you're going to see it more robust. It's going to be more of a dense irregular connective tissue. You'll see bigger blood vessels, right? Up here, in the papillary region, right, you're also going to see, well, you're not necessarily going to see it, but you know it's there, that there's possible shunting between, blood vessels reaching the surface, of the papillary layer, or being away, right? And it's not that the blood vessels are moving, it's that the blood's being shunted either closer to the surface or further away from the surface for, it could be emotional reasons, if you're blushing, it could be temperature related, it could be, like a fear reaction. There's all sorts of reasons why blood might go closer to the surface or away from the surface. So that's the kind of some major things that you're looking for, right? So again, this is the same slide that I showed before. So when you're looking at this, you want to be able to pick out those layers, right? You've got the 5 layers of epidermis in this case, right? Because if you look at the thickest, if you look at this here, starting from here and go up to the surface, ask yourself which is the thickest stratum, right? If the thickest stratum is spinosum, then you're looking at thin skin. If the thickest stratum is going to be the corneum, you're looking at thick skin, right? So in this case, this one here is the corneum, and that's a lot thicker than this layer here, which is the spinosum, right? So if you go through the layers in order, right, you can see here there's a lot of squames, corneocytes that are just flaking off. If you go down to this kind of lighter purple layer, that's where you reach the lucidum. So stratum corneum, down to here, and then you get this light colored layer, that's stratum lucidum, and then you get this very dark layer, and that's going to be the stratum granulosum, and then you get this kind of thick layer with lots and lots of nuclei. If you zoomed in real close, you'd be able to see little evidence of desmosomes, and that's the spinosum. And then if you look at the very bottom here, you can see these are kind of columnar, and that's stratum basale, right? So that's what you're kind of looking for. This kind of pink line that you can kind of sort of see, that would be the basement membrane, because this bulge here and here and here, these are all dermal papillae, right? So you're in the papillary layer of dermis at this point. And if you look at some of these things look like capillaries, little knots of capillaries. Some of them, if they're like an oval shape, like this thing looks like it might be an oval shape, that might be a Meissner's corpuscle. The only place that you'll find a Meissner's corpuscle is in a dermal papilla, right? You won't find it anywhere else. And if you look for them in thin skin, you can look, but they're insanely hard to find, right? So in thick skin, you've got a fighting chance. So that's what you're seeing there. The next kind of division is a bit more subjective, right? So if you look here in this sort of area, and you look at the pink lines, so you see the pink lines are a bit thinner than they are here, right? So this is going to be more of an areolar connective tissue, and this is going to be a dense irregular connective tissue, right? Because these fibers are much bigger, more swirling. These ones, it's more like just thin pink lines kind of doing their thing, right? If you're up here, you might see little bits of capillaries or these Meissner's corpuscles. That's classical papillary layer. If you're down here, right, you'd be expecting to see bigger blood vessels. You might be seeing ducts of sweat glands, that sorts of thing, right? But this is the dense irregular. This is reticular, right? So you've got corneum, lucidum, granulosum, spinosum, basal, papillary, and reticular, right, for those layers. And so any any skin slide that you look at, that's one of the first things you want to do is work out where the epithelium is. Can you identify the layers? That's what you want, right? This one here, the lucidum, sometimes doesn't show up very clearly, but the thickness of the layers is what will give it away. Also, the complete and utter lack of anything hairy is probably a good sign too. Because even if you lasered away every hair that you could possibly see, there's still probably some evidence that there was hair there at some point under the surface. So, because you can only really destroy what you can get to. All right, so that's what we've got here. So you can see this dense irregular connective tissue. You can see luminal structures. So if you look here, you can see a big luminal structure. So that's going to be a blood vessel. You'd expect to see smooth muscle in the wall. You'd expect to see endothelium on the inside. And inside the lumen, you'd expect to see red blood cells and white blood cells. Right? So blood vessels, they vary in form quite a lot, but they always occur the same way, so they're not really that hard to recognize when you look. Like there's another one over here, right? That sort of thing. The reason that's important is because you want to be able to tell a blood vessel from a gland, right? You see these little patches here, like this, and this, and this, and this, and this, right? These are glands, right? They're eccrine sweat glands. How do I know they're eccrine sweat glands? Because the only type of gland that you'll find in thick skin is an eccrine sweat gland, right? You don't find apocrine sweat glands. You don't find sebaceous glands. So you can pretty quickly narrow down what your options should be. So again, if you see the papillary layer here, and you see the reticular layer here, you see these little purple structures, like this, kind of perpendicular to the surface. Right? So those are going to be sweat gland ducts. Right? And the reason they're just in little patches like that is because they're simple coil tubular, and so they're going in and out of section as they go through the skin. Right? So you're never going to see like a beautiful winding coil, because these slices are like, maybe 10 microns thick. You're not going to be seeing a whole lot of swirling like that. Right? But if you see one of those ducts, you can kind of follow it kind of down, and you can make a pretty good bet that there'll be a gland secretion area real close, right? And so you should see, cuboidal cells. So stratified cuboidal is what you'll see with the duct, right? When you get down to the secretion areas, you'll see lots of rings of cells. You might see myoepithelial cells on the kind of outside of those secretion structures, right? But they always occur in these little patches like this, right? If you've got a hose pipe and you balled it up and then you section through, you'd see lots of little circles. That's what you see. All of this white here is adipose tissue, right? So you're going to get patches of areolar connective tissue in between lobules of fat, because blood vessels have to get through, right? And the blood vessels are going to be in areola more often than not. So again, just to make it clear, Meissner's corpuscle in the dermal papillae. Pacinian corpuscles can be pretty much anywhere in the reticular area, but they're usually found deeper in the reticular area, sometimes even poking into the subcutaneous tissue. Pacinian corpuscles are also found surrounding some of the visceral organs, because they're deep pressure detectors, right? Tactile cells and Meissner's corpuscles, they're for light touch, right? You've also got, around the bottom of the hair, you've also got a root hair plexus, which is a sensory network. So even if something doesn't touch your skin, but it changes the orientation of a hair, you can still sense that, right? Like breeze. It doesn't technically like really touch you or like deform the skin in any way, but you can feel it. So that's what you've got there. And again, you can see these glands here. An apocrine sweat gland sometimes enters into a follicle. An eccrine sweat gland doesn't. It just goes straight to a sweat pore at the surface. Sebaceous glands, they're always found. So if you look at the distance of a hair follicle, it's usually about halfway up, and they're always as lobes, right? So it's actually kind of tricky to see the actual duct piece, because the duct piece would just be at the kind of entrance point where it joins with the follicle, right? And like when you pluck a hair, you don't pull out the entire follicle, right? You're only pulling out the kind of inner piece of the follicle at best, right? So, and different hairs are going to be at different stages of growth. So, you know, like club hairs have lost connection with their blood supply, you know, so they fall out easier than growing hair cells and tissue. If you look at this one, so would you estimate this to be thick or thin skin? Thick, right? If you look at the little picture here, you can see there's this big kind of red mass, and then you get down to the purple. So the kind of red staining part, that's actually going to be the stratum corneum. This darker layer here, that would be the kind of, you know, lucidum and granulosum kind of together. They kind of, it's so stained that you can't see anything different, but you know, the red would be kind of where you'd expect the lucidum to be. All of this is spinosum, all of this very, very cellular piece. And these white lines here surrounding the cells, that's where you look for those desmosomes. So if you zoomed in on this image, you'd be able to see those little things. And then these pieces here, so this is a epidermal ridge, and then this is a dermal papilla. And this oval structure here is a Meissner's corpuscle. Right, so the Meissner's corpuscle, it's an oval-shaped structure, and it's got like a little capsule. That's why they're called encapsulated sense organs. Right, so it's got a little capsule, connective tissue capsule, and then inside of it, there's lots of lines going side by side, like this, right, and lots of little nuclei. So the nuclei are going to be the nuclei of Schwann cells, and what you're looking at is like little bits of myelin kind of wrapping around a central nerve fiber. But you're not really going to expect to see the nerve fiber coming out because it's just like a single nerve fiber. So you're not going to see that. All the kind of pinkish stuff here is going to be collagen bundles mostly. You might see like this thing down here. This, it looks like a ring. You see that? That's probably a blood vessel, like a capillary. So pretty small. Capillaries of this type, later we'll call them continuous capillaries. They're only just big enough to fit like blood cells through, right? There'll be different types, like in the bone marrow you saw sinusoids. They're capillaries, but they're big capillaries. Right? You can fit lots of blood cells in sinusoids. But for these ones, they're not very big. So when you see them, they look cute. They've got a little ring. You might see an endothelial cell. You won't see any muscle. The bigger blood vessels, you will see muscle. In this slide here, so this is a finger. Right? You can see the bones. There's a bone here, and there's a bone here, and there's a bone here. Right? So you can see the bone marrow on the inside there. You can see this is a fetal finger, so it's not finished developing yet. So you can see the end still has the hyaline cartilage, which will turn into the articular cartilage. You can see bits of joint capsule, the joint space. Right, so a lot of the same tissues that we identified before you can recognize on this. Right? But what we're looking at here is the fingertip. So at the fingertip, you can see you've got thick skin on the edge. So most of this dark area is stratum corneum. The red piece, stratum spinosum. The very bottom would be stratum basale. you can see dermal papillae. If you're going to find mycinous corpuscles, they're going to be in these little bulges. These big things down here, those are the Pacinian corpuscles, right? So there's one here, 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 and here, right? So another name for Pacinian corpuscles is lamellated corpuscle, right? And the lamellated corpuscle is called because it's got lamellae, right? It's like an onion. It's got layers. And again, at the center, that's where you'd have the nerve fiber. You've got lots of like modified Schwann cells and things going on. So Pacinian corpuscles. And they're actually really deep. They're in this hypodermis, right? Because the blue is going to be the dermis. If you look up here, you can see the light blue. That's papillary part of dermis. If you look at this thicker blue here, that's going to be reticular. You see these red balls? So those are the secretion parts of sweat glands, eccrine sweat glands, right? There's a whole line of them along here. And some of them you can see they've got little ducts coming off. So if you zoomed in on that, you should see it for each of those ducts, it's a stratified cuboidal epithelium. And you should be able to pick out some other round structures. So even without zooming in, you can pick out round structures that look like they have some sort of reddish material on the inside. That's going to be a blood vessel. So those are the common things you're looking for in these pictures. So I mentioned follicles already. I mentioned that follicles are epidermal-derived structures that are buried in the dermis, right? And they need to be buried in the dermis because they need a blood supply for the stem cells, and epithelium is avascular, so it doesn't have any blood vessels, right? So what we're going to do now is kind of look at some of these layers that make up the follicle, right? Because when you see slides of hair in skin, you'll either see it in cross-section, so it'd look like an artery target, or you'll see it as like this in longitudinal section, right? Sometimes you'll see them in oblique, but it's those two extremes that I'll mostly be asking about, right? So you can recognize the individual layers, know which parts are part of the hair, which parts are part of the follicle, that sort of thing. So I went through these things already. For the follicle, again, there's going to be epithelial pieces and dermal pieces. And this term adnexa, you don't see it too often these days. It used to be more common, but it refers to this kind of, the skin and its kind of accessory structures, which, again, for humans is mostly we're talking about hair and glands. Okay, so these are the main layers you're looking at, right? So for the actual hair itself, like if you grabbed onto a hair and pulled it, what you'd be pulling is a three-part structure. So in the very center, you've got something called a medulla or medulla, right? And that's going to be the kind of very central piece. Outside of that, you're going to get a thicker area called the cortex. And then on the outside of that, you're going to get a very thin piece that's going to look like shingles on a roof, and that's going to be the cuticle, right? So the cuticle can become damaged, and that can lead to things like split ends and stuff, right? So, so that's what you've got for the actual hair itself, right? These layers are the follicle, right? So there's an epithelial part to the follicle, and there's a connective tissue part to the follicle, right? The epithelial part is more complicated, right? And so you've got cuticle that's part of the hair, and then you've got cuticle, which is part of the follicle, right? And they're kind of up against each other, sort of like that, right? And so the epithelial part of the follicle, you have an internal and an external part. For the internal, you have the cuticle, and then you have a layer called Huxley's layer, which is usually a couple cells thick and got a bunch of granules in it. And then you get on the outside of that, you get a layer called the Henle's layer, which is usually 1 cell thick. So if you see something that looks like simple cuboidal in the middle of like a hair, it's the Henle's layer, right? And so those are all smushed up together, and then you get a thicker layer on the outside, and that's called the outer or external epithelial root sheath. Is this making sense so far? It's a lot of parts. It's really good when you're reviewing this stuff to draw a bunch of rings and try to name the layers from inside to outside, right? As opposed to just making a list, right? So you've got the internal root sheath and you've got the external root sheath. When you look at a slide that has a bunch of, like a thicker kind of area with a bunch of cells and stuff, that's going to be the external root sheath, right? Outside of that, you'd see the basement membrane. That's going to be called the glassy membrane, right? And that basement membrane separates the epithelial piece from the connective tissue part, just like the basement membrane does for the rest of the skin, right? So here they call it a glassy membrane. It's not really that thick. It's there. And then on the outside, you'd have the connective tissue or dermal root sheath, and that's up against the rest of the connective tissue, right? But it usually shows up as a kind of distinct directional region outside of the hair. When you look at real examples, it makes it look a little bit easier sometimes. I love this diagram. It's got all sorts of like stuff going on, which is like great. But what you want to be able to do is kind of count from inside to outside and name the layers like I just did, right? So you've got the, in the very center, you've got the medulla. So this is looking at the hair bulb. You can see the hair papilla with its connective tissue cells inside of it, bringing blood vessels and that sort of thing. Right, so the medulla is the innermost part of the actual hair itself. Right, the cortex is outside of that, and then you've got the cuticle of the hair. Right, so when you touch a hair, you're touching the cuticle. Right, the rest of this is going to be follicle. Right, so if you look at so you've got the inner, and then you've got the outer epithelial root sheaths, and then you've got the dermal root sheath, right? And the internal root sheath has those kind of 3 parts, right? It's got its cuticle, it's got the Huxley's, and the Henle's. I haven't found an amazing way to remember which ways Henle's and Huxley's goes. I've just gotten to the point, remember that Henle's on the outside. So if you remember that, you're set. I wish I had a fun mnemonic. I have a new fun mnemonic for you later in the lecture, though. So measure your expectations, but it's a good one. That's all I'm saying. So don't want to oversell it. Matrix cells are going to be just the stem cells that are producing the new hair and pushing that out. So this is a really nice example of kind of how that looks. You can see 1, 2, 3. This is the actual hair itself, medulla, cortex, cuticle. And then you can see there's the internal, external, epithelial root sheath, and then you've got the dermal root sheath on the outside. Right? So if I had a bunch of arrows pointing to different locations saying which of these would be an appropriate place to put the glassy membrane, you'd want it to be between the external root sheath of the epithelium part and the dermal root sheath, right? Because it's a basement membrane. These pieces over here about the growth phases, it's just to illustrate that, as hair develops, it doesn't live forever. It kind of becomes a club hair eventually, loses its blood supply, and just kind of drops out of the follicle to be replaced by a new follicle, right? Which is why electrolysis and permanent hair removal tends to focus on damaging or disrupting the follicle and the stem cells in some way. Otherwise, it's just pulling out the hair or trimming the hair. these terms up here, these are different forms of melanin. So, some people have like purely black hair, some people have like purely blonde hair, and some people have like something in between, right? But it's all ratios of which type of melanin you have. So, eumelanin versus pheomelanin for the two different sets of pigment. All right, I think that's all the new stuff. The rest of this is just kind of repeating what you've seen on other pictures, so you can look through those. But longitudinal section here, cross section over here. So, and I've got some examples that you can see for those. So if you look at this, you can see a hair follicle over here that's in oblique section. You can see a hair follicle over here and here and here, which are in longitudinal section. Right, so you can see the bulb at the bottom. You see this light area? That's going to be the hair papilla with the connective tissue part. If you see yellow on these slides, it's going to be the actual hair root. Right, so the hair root is inside the follicle, and then you can see the different layers of follicle outside of that. Right. If you look for, so you see this very, very skinny red thing here? See that little skinny line? That's an erector pili muscle. If you zoomed in on that, you'd see it's a bunch of smooth muscle cells which are just moving down towards the follicle. Right? Sometimes they look a little bit thicker than that. Sometimes they look exactly like that. Sometimes you just see patches because it might be of an erector pili that's kind of going in a different direction. Right? This one, it's caught pretty much perfectly, so you can kind of see the whole thing. This gland here, and here, and here, and here, right? All of those little, blobs, right, of cells that if you zoomed in on them, they'd look like they were filled with bubbles, right? Those are sebaceous glands, right? They're always going to be kind of up, attached to the side of the follicle, right? When you see little patches like this, or this, or this, where you see some lighter and some darker tubes, that's going to be sweat glands, right? Most likely equine sweat glands, because unless you're specifically taking a section from the armpit or the groin, you're not really going to see apocrine sweat glands. Right? It's much more of an intentional sectioning going on there. Right? But for scalp or just any other area of skin, it's going to be eccrine. Right? And so the darker areas are the ducts, the lighter areas are the secretion areas. Right? So you can see a bunch of those good examples on here. For the layers, you can see that the stratum corneum is very thin and very wispy. The thicker tissue is going to be the stratum spinosum. Right? There won't be any sign of a lucidum. This lighter pink is papillary layer. This darker pink area is dermis. Right? So even though I'm kind of using low power zoom on here, you could easily go to the slide itself and find all of those things in like glorious high resolution. Right? So any of these things that have a lumen like this, pretty much blood vessels, right? If you zoomed in higher resolution, you'd easily be able to pick out capillaries too, right? So they're all there. Some of this deeper tissue, that's getting into like the denser, deeper fascia, and some of the even bigger blood vessels. So again, when you see secretion parts of glands, you can look for the actual secretory cells with the lumen. You can look for myoepithelial cells. You can look for duct cells, right? These are all exocrine glands. Sometimes you'll see this term sudoriferous. It just refers to sweat glands, right? There's the oil glands, which produce the sebum. That's sebaceous glands. There's the sudoriferous glands, which produce the sweat, and they're either eccrine or apocrine. There's a close-up of what I just showed you. you can easily see eccrine sweat gland here, eccrine sweat gland here, lighter secretion areas, darker duct areas. Over here, this round piece and this piece here, sebaceous glands. This is that nice erector pili. But if you look over here, you can see a patch of smooth muscle too. That's another part of an erector pili, of a different one, right? When you look at skin, you're looking at dozens and dozens of different structures all kind of packed together. And you can see layers of follicle. So it's hard, you can't really see the actual hair root in this one. It's just an empty follicle. But you can see there's like, cellular areas, and then on the outside you can see there's a pink piece. So the pink piece here on the outside would be the dermal root sheath. On the inside, the cellular piece is the epithelial root sheaths. So again, same thing, just spelling it out, what's happening. If you looked with a transmission electron microscope image, which I haven't included any here, but you could easily find them, you can actually see there's organelles, like we looked at in Unit 1, inside each of these cells. Right? The secretory parts of the sweat glands are kind of complicated. A lot of basic textbooks just call it simple cuboidal epithelium, right? And then the duct parts are stratified cuboidal. But anyone that's got eyes that's ever looked at one of these things, right, can see that it looks like a hot mess in there, right? It's not just a simple, nice arrangement of square cells, right? An apocrine sweat gland looks a lot more like that. For the eccrine ones, it's always just a messy pile of these things. Some look more like simple cuboidal, but some definitely don't, right? I think that might be a nerve. Isn't that cute? It's like seeing a baby penguin. It's like, no, nerve. No? Okay. You'll get there. Don't worry. It's coming. And then it's like you've gone too far, right? Anyway, but when you look at these, right, there's actually multiple cells kind of in there, right? And it's super hard to tell, like, these dark ones from these light ones, but they're in there. And so you just want to recognize the light staining secretion areas from the darker duct areas, and the darker duct areas are going to be stratified cuboidal, and that they look nice. So you can easily recognize those, like this. Myoepithelial cells, when you see those are going to be around the outsides of the secretion areas, kind of close to the basement membrane. And the basement membrane, when you zoom in real close, actually looks pretty prominent. Should be a nice pink layer. Okay, so this slide just says hairy skin, but it's actually hairy skin from the armpit. And the reason I know it's probably from the armpit is because histologists tend to section the armpit preferentially over the groin for some reason. Finding pictures of groin histology is not as easy as finding armpit. So just so you know. And when you look at this here, you can actually see there's some evidence of pigmentation in the lower part of the epidermis here. You can see that the stratum spinosum is thicker than the stratum corneum, so this is thin skin. If you look here, you can see this is a hair follicle. So it's not a beautiful one, but it's definitely one, right? So you can see that there. And then you see this giant thing here, all these giant holes, right? So these are lumens of an apocrine sweat gland, right? And to show you how different it looks from an eccrine sweat gland, this is an eccrine sweat gland. This is an eccrine sweat gland. This is an eccrine. All of these small tubes are eccrine. This monster over here is apocrine. Right? So when you see them side by side, they look nothing alike. The lumens are much, much bigger. In the apocrine ones, the whole gland is much bigger, and it's going to be associated with a follicle. Over here, you can see this kind of round thing that's got a bunch of bubbles. That's going to be a sebaceous gland. So if you zoom in, again, you can see that these look simple cuboidal. They look great for the secretion areas. The duct part's going to be right up against the follicle, so it's actually harder to see in an apocrine gland than it is for an eccrine sweat gland. For an eccrine sweat gland, you can see these very messy looking secretion areas, and then you'd be looking for areas like this where it's got a double layer of cells, and that's going to be a duct. Right? So the ducts all start in the secretion areas and then travel to the surface. This tissue is dense irregular connective tissue, so lots and lots of irregular collagen bundles, fat cells. Over here, you can actually see a rocket ship looking thing that's actually a hair follicle with sebaceous gland lobes out to the sides. The yellow thing is actually the hair root, and then the purple is the actual follicle. Some other, again, rare ones that you're going to see, like it's not super duper common that you're doing histology of like ear canals unless you're specifically looking for something, right? So, you know, the wax glands are modified apocrine glands, right? So you can see here they look sort of like the apocrine glands, like this. Looks similar to this, big lumens. But instead of producing just sweat, it's producing a thicker secretion, cerumen, which is wax. So again, same sort of thing here, looks a bit different. So some other areas that are of interest, nails and toes, right? So we don't have claws, right? Most animals have claws. Primates have nails. We have these flattened things, right? So there's certain parts of that nail that you want to know, right? So when you're looking at the kind of gross anatomy of it, you've got this kind of overhang, which could be long, could be short, depends how you cut your nails, right? But that overhang there, that's called the hyponychium, that space below the overhang, right? The eponychium is on top at the back, right? So you've got the flat piece of the nail is the nail body or the nail plate. At the edge, the free edge overhangs the hyponychium. So if you get dirt or something trapped in that space, it's in the hyponychium, right? The cuticle, so technically eponychium and cuticle are two slightly different things, but most books just call them equivalent things, right? And so this kind of edge towards the back, that's where you're going to get the eponychium, right? Again, lots of variation. Some people have really nice half moons at the base of their nails. Some people don't. Not wildly important. But when you see that, it's called a lunula, right? And it's just a kind of thickened area, right, of, you know, underneath that area. And nails, when you push on the nail, you kind of compress it, you probably see that it goes kind of lighter in color, and then the kind of red color kind of returns, right? So you're kind of pushing the blood away, and then the blood returns, right? So it's a way to check that you have circulation. Look at that. And that's because the nail is transparent, right? Underneath this, so in this area here, that's called the nail root. That's where you get the matrix cells, where the new nail is produced and it pushes out like that. And then you get these lateral margins, which sometimes grow nicely, sometimes they can curl in and create ingrown nails, which is not a fun experience. I can tell you that it's not a fun experience. But this is the lateral margins. You got the hyponychium, eponychium, and the nail root with the nail plate body. If you compare that image to this image, you can see the same thing, right? So on the palm side, what the fingerprint would be, that's the thick skin, right? And when you look at a fingertip, right, what you'll notice too is there's lots of little lines, right? So those fingerprints, those are called friction ridges, right? And the friction ridges are a great place to look for sweat pores. So if you zoomed in on those in a really high detailed picture, what you see is a lot of pores in between those friction ridges, and that gives grip, especially when the hand is wet. This shape here is just a deformation of the tissue because of the preservatives that were used. They didn't have a really, really funky fingertip. It's just that it got deformed. But you can imagine that being kind of more normal. If you zoomed in on that, it would look like typical thick skin. Over here, it would look like typical thin skin. Right? You can see the various parts here: eponychium, hyponychium. The nail bed's the part that underlines the nail plate. Right? So the nail bed's got lots of blood vessels. The nail plate is the keratin structure, very heavily compacted keratin. PH stands for phalanges, so it's a phalanx, distal phalanx, if you're into anatomy. So, and if you compare it to this image, it's the same sort of thing, right? You can see the phalanx right here. You can see the thick skin and the actual nail itself. It looks very kind of fragmentary the way it's been cut. But that's where you'd get the nail plate, the nail bed, and the nail kind of root, and the eponychium. Hyponychium is almost non-existent on this one, but it's there. All right, so getting to the next kind of interesting region. So we did the eye in the context of, for the last unit, we did the retina, right? So the retina is the photosensitive part of the eye internally at the back there, right? And we looked at the layers. Right, so now we're going to go through some of these other parts of the eye, which are interesting. Eyelids are very interesting areas of skin. So they still have, hairs, but the hairs tend to be concentrated at the ends as eyelashes, right? Then that would be kind of cool, right? If everyone had like really, really hairy kind of carpeted eyelids. That's haunting. Good. That doesn't happen, so that's good. But when you look at a picture like this, you want to be able to tell what's the outside and what's the inside, right? So the outside is going to look like regular thin skin. So if you look here, SK for skin, you can see it looks like regular thin skin, and you should be able to see things like, all the regular layers that you'd see. Over here, TG, these are called tarsal glands, right? So the tarsal glands are going to be the glands which are on the inside of the eyelid, right? And they're going to be producing like a tear film, which is going to be keeping the eyes kind of moist, right? And what you're going to see is this kind of very, very, very innermost layer of that is going to be called the conjunctiva. There's actually a layer that covers the eyeball, the white part, not the cornea, the white part. And then there's a part which covers the inside of the eyelid, right? So the conjunctiva is kind of covering both. If it's covering the eyelid on the inside, it's the palpebral conjunctiva, PC. If it's covering the eyeball, it's the bulbar conjunctiva. I mention that because they're different. I actually re-uploaded this PowerPoint last night because I was like, I need pictures of the conjunctiva, because instead of just telling you about it and describing these different shapes, I actually included some pictures. So hopefully the slides make a bit more sense now. But yeah, and you've got multiple sets of glands on these things, right? So tarsal glands are on the inside. Tarsal plate is the connective tissue, kind of like foundation of the eyelid that the muscles are attached to. So like OO is orbicularis oculi. That's the muscle that's kind of going around the eye to close the eye. There'd be another muscle called levator palpebrae superioris that helps you open your eye, right? But those are skeletal muscles that you can control, right? Connective tissues, your tarsal glands here, which are different than your lacrimal glands, right, on the outside of the eyeball. Down here at the tip, you've got the ciliary glands, right? So eyelashes are also called cilia, which is awfully confusing because they have nothing to do with cilia, the cellular structures, right? They're just called cilia, right? But eyelashes have little glands at their base, right? And there are two types. Here's my mnemonic, right? You'll love it. right? So these glands here, glands of Zeiss and glands of Moll, right? I want you to be able to remember if these are modified apocrine glands or modified sebaceous glands, right? Because you'll be able to see both down there, and you want to know which is which. You know the ice cream, Häagen-Dazs, right? So how do you spell Das? It's a fun question, right? Question 1 on the test. I'm just thinking about making the first portion of the test entirely about Häagen-Dazs ice cream. I love my little fantasies. D-A-Z-S, right? That doesn't necessarily work for our mnemonic here, but Das rhymes with mass, and mass, if you can imagine Häagen-Mass, like a type of I scream, layers, M-A-Z-S. Moll is apocrine, right? Zeiss is sebaceous. That was totally worth it. Come on. M-A-Z-S. You don't want to remember it, but you will. All right, so that's what you've got there. So these ciliary ones, the tarsal ones. This is another really good picture. You can see some muscles, skeletal muscle. You can see the tarsal glands. You can see folds for like the conjunctiva. All the different parts like iris, lens, all those things, right? This relates to what you probably know from general anatomy. For this, you don't need to know a lot about the lens. What you mainly need to know is that on the outside there's a lens capsule, which is essentially a basement membrane, right? And from there you've got these square cells, the lens epithelium, and that's producing these new cells which are pushing towards the inside, creating this compacted mass of crystalline-filled cells, which you call the lens, right? And so you get these developing ones, or differentiating lens fibers, and then you get the mature ones. They're filling with crystals as they go. For the cornea, it looks really different. You've got a front side, you've got a back side. Confusingly, there's a Bowman's membrane, and there's a Desmet's membrane. The Desmet's one is actually a basement membrane. The Bowman's one just looks like a basement membrane, right? So here you've got this stratified squamous epithelium, right? And then you're going to get the, there's a basement membrane there, but it's not called Bowman's layer. And then you get this connective tissue, stroma, and then you get Desmet's, right? So it's like, and when I say cornea, I mean the transparent front part of the eye, right? The stuff to the side, the white of the eye. The white of the eye is covered by conjunctiva. The cornea is not covered by conjunctiva. Conjunctiva, here, if you look at this, it's got two different types of histology. This piece here is a stratified columnar epithelium with tons of goblet cells, right? This, more covering the sclera, is going to be more of a stratified squamous epithelium that doesn't have goblet cells, right? So they're both conjunctiva, but very different types. I've tried to spell it out nicely there. And the last thing is, in addition to the tarsal glands and the ciliary glands of the two different types, you've also got lacrimal glands. These are pictures from the textbook. I think they look okay, right? You can see a lobe. You don't see a lot of information there, right? You can see it's like got kind of acini. It's actually serous cells. They look kind of light, like you'd expect from mucus, but if you look close, they've actually got lots of light staining granules, like serous, right? So that's what you see there. Same here, very complicated glands. This is a much better picture. You can actually see duct areas and secretion areas, blood vessels, all sorts of good stuff. And that's good. If you need to practice, I did post an additional set of skin images. Oh, ho, ho. Even some eye ones. Look at that. Love that. I'll get the grades posted this afternoon. So if you have any questions, let me know. Also, if you have quizzes, I think I fixed all the issues with quizzes. Right? I don't want to get to the end of the semester and have people arguing about quiz points. Not going to happen. It probably will happen, but it shouldn't happen. Let's not let it happen.